Anyways, I'm going to talk about drug delivery and CRISPR, some of its applications, and basically I'm trying to introduce some of the stuff that we do in our lab indirectly, make it fun for you. I hope you enjoy it. This is okay. You're gonna need this one. So before I talk about drug delivery, I want to talk about how drugs are actually developed. The way that drugs are developed, you basically first identify your target. This is so cool, I can see my slide in front of you. You can identify your target, then you do your drug discovery. That means that you look at molecules, small molecules, protein that are that can interact with your target and do whatever you ask them to do, basically. And then you start optimizing your target, because you're not going to have one type of drug. You're going to discover a lot of different types of drugs, and then you want to make sure which one is actually working the best with your target molecules. And then you find your lead compounds, and your lead compounds can be only one or two molecules at most. And then you start optimizing those lead compounds based on your needs, how you want them to get to the cells, what you want them to do in the cells, what type of a tissue you want to deliver them. And once you get those things optimized in terms of formulation and delivery, then you start your preclinical testings in cells or in animals, and then you're going for clinical trials. I have many friends who work on drug delivery development, so I wanted to contact them to actually ask them how can I be more effective in talking in a non very scientific like terms. It was kind of hard, so I actually brought the person with me to talk with you about it. So, so I'm going to invite Hector to talk about it, because he's been involved in some of these drug development. So he's going to talk with you about how these things are done in actual industry, and then I'm going to continue.
Okay. Well, I'm not going to go through these slides, so these are your slides, and nice that I just step in. So my name is Hector. I'm a product manager at a device company in San Diego called Cardea, and we've been working with Kiana's lab for some time now. My colleagues Brett and Francie are over there too, here for the day. And my particular background is I had an undergraduate degree in chemistry, a PhD in biophysics. I did a postdoc in yeast genetics, and for about close to 10 years I was in drug discovery at different big pharma and small biotech companies, focusing on antibody drug discovery and development, mostly for cancer. And so I want to tell you just a little bit about what goes into drug discovery before we talk about the delivery systems, which Kiana will do a little bit more of. There are two basic kinds of drugs. There are large molecules, antibodies, and there are small molecules, okay? And I'll tell you more about the large molecules. Those are the ones I have the most direct interaction with. So as Kiana was mentioning before, you begin any drug discovery campaign with understanding what you're trying to target. Perhaps you understand that in some kind of cancer, some protein receptor on the surface of the cell is vastly overexpressed, and that thing is driving cancer and cell propagation. So you want to shut that down. And you can do that in a pretty straightforward fashion with antibody discovery engineering technologies. And there are three big steps. First, see if your antibody works in a test tube. Then see if your antibody works on cells that you're growing in a petri dish. And then finally, see if your cells work in some sort of animal. So that's all before you get to the clinic. And these have become actually fairly mature technologies. I think the first FDA-approved antibody was sometime in the late 80s, early 90s. And since then, it's just ballooned, and everybody's learned better and better ways to make antibodies. It's really turning the crank. If you know protein X is expressed on your cancer, there's going to be a dozen companies going after it really fast. There are lots of great ways of coming up with antibodies, either with technologies in a test tube, or injecting a mouse and just extracting the serum, or injecting goats or cows, or all sorts of things. It's pretty cool, the sort of things we can do with protein engineering now. So in the first step, you take all of these, you screen them in a test tube, find those that bind very tightly to your protein of interest, and then take those forward. In the next step, now you have some cells growing that are from your cancer of interest. And you take those that came out of the first screen, add those to your cells of interest, and see if any of those also arrest cell growth. Hopefully some will. And at that point, you start going into more in vivo models. Usually the simplest the simplest thing to do is to artificially grow a cancer in a mouse. So take some of these cells that you grew in a petri dish, inject them into a mouse, the mouse grows a tumor, now you put your drug in there and see if the tumor shrinks. It's really that simple-minded. Like you're literally measuring the size of the tumor over time, over weeks. And then you go forward. Usually people do safety studies in primates to make sure it's not going to hurt humans when it gets there. And if all this looks okay, then people are usually ready to make the big investment, financial and otherwise, to go into clinical trials. So I mentioned, it wants to bind tightly, it's having this activity in the cell and everything, but the other considerations before a drug goes forward that I didn't mention is, can you make a lot of it? Can you make a lot of it cheaply? And is it going to be stable on the shelf? So it's really a multi-parameter problem that you have to keep in mind from the very beginning. So you're always testing multiple things and keeping going it forward. And then, as I said, all that looks good. You get to patients, you identify the right patient population that might be overexpressing this protein of interest, and hopefully it'll help some patients in the clinic before they go forward even further. Let's see, I think there was one more thing I wanted to share about that. Antibodies, drugs. Well, just the final thing I wanted to point out is I mentioned that these technologies have become very robust. It's actually very, very easy to come up with an antibody for basically anything you want. So, in my opinion, what's driving novel drug discovery and new efficient ways of stopping disease with drugs is two things. One is understanding the biology. Like, do we really understand what's a good thing that's going to shut down the cell, or what combination of drugs are going to shut down the cell growth? And two is, how are we going to get the drug to the cell? It could be in the brain, which is notoriously difficult to target. And with that, I'll back to Kiana.
Okay, thank you so much. I'm not calling again later.
So you heard about all of these steps to actually have a drug and get a drug to the market. But then, in which stage of this development do you think drug delivery becomes important? That's a question for you.
Beginning.
Okay, we have one beginning.
The middle or the end?
Actually, drug delivery is important in every single step throughout development, because when you're identifying your targets, right? If your target is a brain, or if the target is your small intestine, if the target is a tumor somewhere else, you have to actually design drugs that are appropriate for that. You have to make sure that the drug actually gets to the target tissue. When you're discovering a drug, is it actually feasible to have a drug like that? For example, if you're trying to treat something in the small intestine, for example, and you find a biological compound that actually treats that region, for example, in the small intestine, but it's extremely toxic, can you actually give that through injection, where it actually goes everywhere in your body, or you want to deliver it locally? So you have to consider those parameters. And in optimization, when you're optimizing it, it's also optimizing its delivery. How do I make sure that I get enough of that drug to my target cells or target tissue? And this also becomes a delivery problem. And in your preclinical testing, if I'm testing it in small animals, how do I deliver this drug? If I'm testing it in human, can I use the exact same thing? For example, if you're designing a capsule that is supposed to be disintegrated in the small intestine, and you're using a rabbit animal model to study that, is that the right model? Because rabbits are grass eaters, so everything that you give them will stay in their stomach for 8 to 10 hours. Is it similar to human? No, because the gastric transit time in human is about 2 hours. So you can't develop something in rabbits or mice and then give it to human and expect the same thing. And then in clinical development, a lot of times you're developing something and then it goes out to clinical market and it comes back because it doesn't have the same efficacy for all patients from different ethnicity or age. So you have to go back again and improve its delivery. So how do we? This is what I just said. So how do we define a drug delivery system? So basically, I'm reading from here, it represents the science related to getting a drug from the site of injection or the site of administration to the target site. So basically, it can be formulation that, for example, the thing that you get with alveoles or some of the drugs that have a coating that only disintegrate into your small intestine, for example. What is your tissue targeting strategy? Are you giving an injection to a specific tumor? Are you putting an implant for a brain to deliver some sort of a drug to brain? What is your tissue targeting strategy? And what is the intracellular targeting strategies? That means that if you're, for example, designing a drug that's supposed to treat something in cancer cells, how are you going to make sure that that drug doesn't act in a healthy cell? So there are methodologies that are developed. For example, the cancer cells have a different pH inside them. So the people are developing methodologies that they develop a drug, they attach it to something else called prodrug, and that drug is not active unless it's cut and cleaved from the prodrug. So what they do, they create a linker between that prodrug and the drug, and that linker only cleaves or disintegrates at a specific pH of cancer cells, for example. So this way you can make them specific to cancer cells. And methodologies to enhance drug bioavailability. How do you make sure that you get a large amount of drug to the target tissue, the right dosage, and how are you developing basically system for improved patient compliance? So if I'm supposed to take a drug on a daily basis, do I want to get daily injection or do I want to have an implant? So these are all falls into drug delivery science of drug delivery. I want to focus a little bit more about oral drug delivery system because that's kind of I have more expertise in that, and it's I think it's where we're all moving forward. We're not going to have injection hopefully in the near future. There's a lot of work that are being done on that, and we'll talk about it more. That why injection is not the most preferred route of drug delivery. Why do we like oral drug delivery? Why do we like to take oral capsule rather than getting injections? Anyone?
It's easier.
It's easier. What else?
It doesn't hurt.
Doesn't hurt.
No waste. There's like no needles or waste.
Yes, yeah. You don't need to go to clinic, you don't need to have anyone come and give you injections, So it's patient convenience, non-invasive, it's high compliance, and from a manufacturing standpoint, to develop those capsules and pill, we actually have the infrastructure to do this thing. So if you can develop a drug in a powder form that is effective orally, then you have the infrastructure to actually make that. So if that's the case, why is that we don't have everything in an oral formulation? Why do we have to get injection? Anyone?
Is it the molecule? Different size of the molecule?
Yes. And what is different size of molecule means?
Would it be too many? Or they're too large, right? So they have to be...
So I'm gonna...
It could be...
No, go ahead.
I mean, the pill would be huge, right?
Yes, that's one of the
The
problems.
volume, you're talking about like actual volume of drug.
But that can be actually considered as a point too. So when we talk about a small or large molecule, we're talking about small molecules means something super small that it's not basically a biologic. So biological that are taking the world like a storm for treating a lot of diseases, including insulin, Humira, all of these are large biologic protein. What does our oral GI tract do when you ingest something that is biologically active? Digest it. Otherwise, you would take, every single bacteria or every single food that you're eating, you'll get sick, right? So our body is designed to digest everything that is biologically active when you take it orally, right? And this starts from the minute you put the stuff in your mouth. Then as it gets to small intestine, stomach, you have very high, very acidic pH that basically destroys everything. Then you get to your small intestine, and you have this thick mucosal tissue that is not allowing any molecules larger than a specific site to pass through it. So a small molecule gets absorbed faster, but when you take large molecules, they won't be able to get past through the mucosal of your intestine. So there's a lot of problem with that, and so it's been super difficult for we're developing all these like biologically active, super effective drugs like Humira. I don't know if you have anyone in your family that uses Humira. My dad uses Humira, and it actually changed his life. the way that it's treated, it's super, super effective. But the problem with it is he has to go to doctor's office every 2 weeks to get the shots. And then he can't travel. He's like, oh, who's going to give me the shot? If I want to go back to Iran, I don't know where to go. So it's limiting him by, so if we can actually develop this drug orally, that's super effective. So some of these drugs, like Humira, are also used for a lot of patients who have inflammatory bowel disease or Crohn's disease, right? These are inflammation in your small intestine. So now you're giving this immune suppressive drug like Humira by injection. What happens is only a small amount of this drug actually reaches the site that it's supposed to act, and the rest of it just gets everywhere. And you have a total reduction in your immune system. That's why people who are on Humira, they have to be super careful not to be exposed to like sick people, and if they get sick, it's really hard for them to actually recover. So all of these things are problems. Having said that, there's a lot of effort to actually improve this drug delivery system for oral drug delivery. So again, we talked about all these things, and these are all of the problems I listed. When you're taking drugs orally, you have to encounter every single of these challenges before you can actually have the drug absorbed. There's one more thing with the oral drug delivery. So oral drug delivery is effective when you're delivering something, it's convenient, but it's not good for everything, right? So if you're trying to treat brain, something, or some tissue somewhere else, if you take it orally, a lot of this drug is going to be degraded and by your liver, right? Because your liver is basically designed to digest and degrade everything. So oral drug delivery is not for everything, but for the majority of if your focus is patient compliance, the best thing is your focus is delivering the drug locally to the GI tract is of interest. But for other drugs that are the target tissue somewhere else, you may need different delivery mechanisms, such as implants. And I don't have those here, but we are designing, not we, but people in drug delivery systems. They are designing non-invasive implants where you can just put like a patch on your skin, or like an implant which you can put once and it delivers drug over a year. Or implants for back of the eye disease, because back of the eye disease are super difficult to deliver drug to, especially if you're delivering biologics. So those patients, they have to actually get injection into their eyes. Just thinking about it is painful, right? So there are delivery systems that they do a surgery once, they put an implant in the back of your eyes, and they can actuate this drug delivery system to just deliver the drug at a certain time point. So parenteral delivery systems, when you're getting your drug delivered to your skin, there's advantage to that because you have the full dosage delivered, so you're not wasting drugs. Some of these biologics drugs are super expensive, so you don't want to give them orally sometimes, because if the GI tract is wasting 90% of the drug, you're having only 10% of it reaches the target. At this point where the bioprocessing is still a very expensive process, you want to make sure that just give them an injection and make sure that everything is delivered. Again, back to Humira, each of the dose of Humira is about $2,000. So sometimes if the patient don't do it right and then they see the drugs coming out, that's money that is pouring out. It's like $2,000. So it's really an important issue. Not going through that. So, what are we moving in terms of drug delivery? There's a lot of interest in drug-device combination. That means that we want to avoid. There's a lot of things that you can do with formulation. That means, how do I make a polymer that protects the drug so when I swallow it, it keeps the drug safe, and it only releases the drug when I'm in the small intestine. But remember, when you're designing drug delivery system, formulation can be super, super expensive. So a lot of people are trying to actually combine devices and drugs in order to avoid significant amount of formulation. Example of those for oral drug delivery, these are my favorite. We have Philip-I-Pill. So Philip-I-Pill was first designed to there's a capsule that you swallow and it monitors your pH. But then recently they have been incorporating reservoir in it that it also has drugs in it. So basically the capsule, as you ingest it, it monitors your pH, and your pH changes as you're going from your stomach to small intestine. So once the pH goes up from small intestine, from your stomach, from like 3 to 5 to 6, 7, 8, the sensor can actually send you a signal that I'm in your small intestine now, and then you can do it on demand, or the system can itself start releasing the drug only in the small intestine. And there's a lot of other information you can get from this system, because they have temperature monitors where as they pass through the GI tract, they can also monitor the temperature. So there are areas in the GI tract, if you have inflammation, the temperature will be a little bit more. So you can actually from that also predict where you potentially have the inflammation, because it's important for people who have like IBD or Crohn's disease. Sometimes they have to go in there and cut pieces of their small intestine. So this can be a good predictive tool for them to see where they can actually cut the intestine. There is Rani Therapeutics that is developing this ingestible capsule. These capsules it's a needle capsule. So you basically have a balloon that is collapsed. On the surface of the balloon, you have these polymer needles which are coated with a drug. And when you swallow the balloon, once it gets to your small intestine, the balloon will start expanding, and this small needle will get into the wall of your small intestine. So in your small intestine, you don't have pain receptors, so you don't feel it, right? And then as the balloon moves with the peristaltic motion of the small intestine, the needle will break down, and then they dissolve, and then they release the drug inside the walls of the small intestine. Surprisingly, this company has raised over $150 million, and they have a lot of drugs in their pipeline, and they have been very successful for the last few years. Medimetrics was another company which is designing ingestible capsules. So Medimetrics uses a very simple electronic. It's basically a diode that sends a light and receives the light back. As it moves down the GI tract, it can actually say where in the GI tract it is based on the signal, sends light, gets light back, and process that. So you can actually have some sort of a drug in it, and once you pass let's say the pyloric valve or other valves in your GI tract, it can tell you where it is, and then you can on demand have it release the drug. So this is another company. You have Lyndra. So Lyndra is a very interesting company. It's a star-shaped drug, and this is developed for extended drug release. So instead of taking a capsule on a daily basis, you take this capsule for 1 week, and you have the drug for basically 1 week. The way it works, you have the capsule, it's kind of collapsed, it gets to your small, it gets to your stomach, and then in the stomach it opens up. It's like a star shape. So it's big enough, it cannot pass the pyloric valves, it cannot go from your stomach to the intestine. As it's sticking there, it releases the drug very close to pyloric valve, so the drug, as the capsule degrades, the drugs get released to the small intestine, very close to the small intestine. It stays there for 1 week until it gets small enough to be able to pass through the small intestine. So you constantly have this little Lyndra capsule in your stomach. And Sonobio also uses ultrasound to deliver drugs. So cool, right? So get back to Ronnie. Yeah, you don't like Ronnie, guys. I didn't like Ronnie when I was back when I was a postdoc. So I had a technology which was basically initiated from, remember the jet injector for vaccine delivery? Those like jet guns that they use to deliver vaccine. So in my mind, if you have those jet guns can penetrate through the thick layer of the skin, right? Why can't we use it in our small intestine? Because in our small intestine, we only have a single layer of cells. So if we can deliver drugs, vaccines specifically, vaccines are also large molecules. If you can deliver the drugs through the skin, you can develop something that can basically shoot the drug with high pressure in the small intestine, you can probably enhance its absorption, and maybe you can have vaccines instead of shots. You can have vaccines inside your small intestine. So I developed this system that you can see right here. Where is it? Right here. So basically it's a two-part system. You have citric acid in one chamber, and you have sodium bicarbonate in other chamber. When citric acid and sodium bicarbonate mix, what happens? you get carbon dioxide, right? So the system was designed in a way that when it gets to your small intestine, there was a valve between the valve that was separating the citric acid from sodium bicarbonate. When the valve degrades, the two come together, start creating carbon dioxide. And the carbon dioxide is generated, pushes a piston to the drug reservoir, And once it reaches a certain pressure, there was a nozzle that would open up and deliver the drug with high pressure. So this is what we designed. We tested it in rabbits. That was not a good thing. So what did I tell you about drug development? If you test things in rabbits, can you try them in human? So first time when we gave the pill to the rabbits, I'm talking too much, I don't think I'll get to the CRISPR, but anyways, when we gave the pill to the rabbit, so we told the vet that the pill is not degradable, and we're hoping that the rabbit will, excrete the pill. So we gave the pill, and then I was sitting down, poking poop one by one to make sure that when is the pill is going to be released. We could not find the pill. So we sacrificed the mice, the rabbit, and we looked everywhere in the GI tract. We could not find the pill. And the vet was freaking out. She's like, you told me it's not biodegradable, and now it's degrading. I'm like, it's impossible for a plastic system to degrade in like 48 hours. And then we opened the mouth. The rabbit was holding the pill in his buccal area the whole time. And it was so annoying because we did all this studies. We were taking blood from the rabbit, and he never swallowed it, and he managed to keep it in his buccal area. So anyways, when we continued the study, we made sure that we actually gavaged the pill, make sure that the rabbit had the pill, and we took X-ray to make sure that the pill was in the stomach, and we monitored how long it took for it to pass. And then we realized it took the rabbit 10 hours to pass the pill from the stomach to the small intestine. So sometimes the pill will get activated in the stomach instead of the small intestine, because sometimes when you put things in for longer period of time, they're not designed to actually work with those parameters. So that's why I moved and I delivered the drug in the actual buccal tissue. And then it was interesting because when we designed this technology, we wanted to do protein delivery. But then because the studies was failing and we didn't have pig in Berkeley to do the study, we just did the study in the buccal cavity. Okay, you want to hold it in your cheek? Hold it in your cheek. I want to see if I can get some protein in your systemic circulation. So we gave the drugs, and it was basically a protein-based drug. We couldn't see anything in the blood, but what we saw, it was a significant immune response to that specific protein. So basically, we saw antibody generation. We saw antibody generation not only in the blood, but also in the mucosal tissue. So when you get a vaccine by injection, you get antibody titers in your blood. And it's really desirable to get antibody titers in your mucosal tissue. Why? Because this is the first entry for a lot of pathogens that are making you sick, right? So if you can protect right there at the site of the entry, it's significant. And this was unknown. Till today, we don't know how pressure actually recruits more immune cells and activate the immune response. And because vaccine is it's not something that VCs and pharmas are interested in. It's not it's not a money generating technology. But the technology has been licensed by Progenity in San Diego, and they're mixing it with the technology from Metametrics with this one. So they acquired this technology and they acquired my technology, and they're mixing the two together, and they're delivering Humira for Crohn and IBD disease. So hopefully soon these patients don't need to take injection for their disease. Okay, where was the aha moment? My aha moment was that rabbit that was holding the pill in this buccal tissue. So next I want to talk about CRISPR. We have 15 minutes. Okay, CRISPR. How many of you are familiar with CRISPR? Okay, you want to talk about it? You want to say what CRISPR is? Okay. Okay. So CRISPR is a part of bacterial immune response. So I brought Hector because as Hector is trying to commercialize this technology, he has to describe this technology for a lot of people. And I also have Brett that he was going to talk about the path to commercialization, but we have only 15 minutes, right? Okay. So I'm just gonna let Hector take over and talk about how he is trying to commercialize my technology and how he's convincing people that this is a good technology.
I think that's you.
Oh, or breath.
Okay. Depends what your next slide is.
It's this. It's talking, explaining CRISPR and biology. Okay.
Okay. So I came up with these slides a couple of weeks ago as something for the non-scientists within my own company who we thought it'd be good to know. They know what we're trying to do. So first, it's a very short introduction to biology. Maybe this should have been done earlier on, so I'll kind of go through it. And then a very short history of biotechnology, including a definition of biotechnology, and then I'll talk about what CRISPR-Cas9 is. Okay. No, wrong way. Okay. Again, I'll go through this quickly. I think you guys probably understand this. Our body is composed of cells. Within those cells and around those cells are molecules. If a cell was a house, the DNA would be the blueprint. The proteins would be the 2x4s that hold up the house and carry out all the essential functions. And here is a cell on the right and a bunch of molecules around it. And I just want to highlight DNA on the top and protein on the bottom right. Now, what's DNA? I think we've all heard DNA. We probably have a decent idea of what it is. DNA is genetic material. It's stored in the nucleus of a cell. It's usually very tightly packed up in this characteristic X-shaped structure that you can see under a microscope often. And as you unwind it, you see this other very famous structure called a double helix, where it's kind of wound up around each other. And that was discovered in 1953. And that sort of culminated a lot of both biological work and biophysics work. And people saw this structure, and this was really the first time people said, oh my gosh, we can actually understand life on a molecular level. Before that, it was just kind of maybe, life seems incredibly complicated. Then this is one of my favorite quotes. Near the end of this paper where they reported Watson and Crick, reported the structure, they say, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Meaning this double helix can unwind and serve as a template for itself. They didn't have any data for this, but they wanted to plant the flag. So zooming in, what is DNA? It codes for proteins. So DNA is a linear polymer of an alphabet of 4 different molecules, and that gets decoded through a process called transcription, through another molecule called RNA, and finally through translation into proteins. And proteins are also linear polymers now of an alphabet of 20 different chemicals. These are called amino acids. And what are proteins? Perhaps we all know, generally speaking, what proteins are, but they are linear polymers of these amino acids with different sequences. And these different sequences all fold up into unique 3-dimensional structures. And these different 3-dimensional structures dictate the function of these different proteins, okay? So on the right, I have 3 famous kinds of proteins. The top is an antibody, which I mentioned before, this characteristic Y-shaped structure. In the middle is insulin, and the bottom is hemoglobin found in our blood. They're all chains of amino acids that fold into these distinct structures and have distinct functions only because they have distinct sequences. If you were to stretch them all out, they're identical. Let them pop back together and they can sort of do these different things. Now, if you have a mutation in your DNA, that will correspond to a mutation in the protein. And this leads to what we call an inheritable or a genetic disease, something that you pass along to your kids. And a famous example of this is sickle cell anemia, where there's a single amino acid change in hemoglobin relative to the normal hemoglobin that causes lots of problems. I'm going to skip this for time. Okay. Kiana,
Yes,
were you supposed to be done at 4?
4:15.
Oh, okay. Okay, so what is biotechnology? I think it's a word that we all toss around and hear about a lot, but to step back a second. The technology is the application of any sort of technical knowledge to some practical use. Biotechnology is the application of biological knowledge to some practical use. And I just want to point out a few places where it's been important. I mentioned here medicine, like we talked about, but agriculture and energy are also things that might not be obvious that have benefited a lot from biotechnology. A couple of highlights in very, very short history of biotechnology. I think the DNA double helix, like I said before, was the first time we realized we could understand and maybe even exploit the molecular mechanisms of life for something that would be useful. Over the next 15 years, it figured out how that translates into proteins. In the early 70s, and actually throughout the 70s, people figured out how to make designer DNA molecules. They figured out how to put those molecules into bacteria and have those bacteria start spitting out actual human proteins. And that's actually the beginning of Genentech at UCSF as well. In 1982, the FDA approved recombinant insulin. That's what we call the protein that comes from not the native source, but something like bacteria. And that was the first protein that was made this artificial way that was FDA approved. Several techniques over the years came about. PCR is one you might have heard of. And then in 2003, the human genome, the first draft of the human genome was developed. It was released. It was a tremendous deal. It was the cover of all these different scientific articles and journal articles. And but at the end of it, what you had was if you just printed it out, just a stack of paper like this big with 3 billion molecules in a row. You still didn't know what it meant. Since then, there have been tremendous breakthroughs in both the analysis of genomes and the rate at which we're generating new genomes. So they're coming out all the time, and Illumina being one of the key players behind this and dramatically increasing the amount of sequencing that's coming out. So we're really starting to get a lot of information. We're starting to understand better how a DNA sequence corresponds to a person's particular state of health, but also raises a ton more questions. So we need more tools to better understand this. And then in 2012, a technology, which I'll talk about now in a moment, called CRISPR-Cas9 came about, which allows the simple editing of DNA, which I'll describe in a moment. Okay, so CRISPR-Cas9, we mentioned this, I think 1 or 2 people in the room know what it is. In 2005, some bacterial researchers identified, I'll just call it CRISPR-Cas9 for now, as a naturally occurring system in bacteria whereby they could protect themselves from viruses. They actually recognize viral DNA and seek it out and chop it up and leave their own DNA totally fine. And over the next 5 to 10 years, they learned more and more and more how to do this. And then they realized in 2012, we could kind of adapt this to make it something useful. So in 2012, the first publication came out where CRISPR-Cas9 was able to cut DNA at a specific site in a test tube. So now we could edit this giant genome of 3 billion cells and only cut one site in this very programmable fashion. A few months later, another paper came out from another lab across the country where they did the same thing in living cells. The paper in 2012 was done at Berkeley, where Kiana was a postdoc. In 2013, that came from MIT and the Broad Institute. These are ongoing legal discussions between the 2 institutes because clearly this has a huge impact on both basic science and medicine, and everybody wants to be a part of it. And today, it's grown in a very short amount of time into a pretty sophisticated system with lots of applications. And let me just describe one to you now that we're working on at Cardea together closely with Kiana. Actually, Kiana, do you want to take over from here since this is yours? Yeah. It doesn't make any sense for me to talk.
So just to recap, I have 6 minutes, right? Okay. Just to recap, what does CRISPR-Cas9 do? It's a 2-component system. You have the Cas enzyme and you have an RNA that guides where that Cas enzyme should cut the DNA and allow the editing in it. So it's a 2-component system. But the way it does it is first unzip the double-stranded DNA. So our DNA is it's a double-stranded DNA, everything 2-stranded are attached. So it unzips, like if you think of it as a zipper, it unzips double-stranded DNA and it looks for things that are matched with the RNA that is guiding it. So basically we program that Cas enzyme with an RNA molecule and we allow this whole complex to go inside the genome and search for it, and then once it finds its target, it cuts it, right? So what is the first thing that CRISPR does before it actually able to edit the genome? It searches, right? It searches the genome. So the search function of the CRISPR is one of its main functions. And so for me, it was like, okay, can we actually use this search function in a way that we can actually look, basically Google the genome? Can we actually Google our genome to see where specific the gene is, instead of like sequencing the whole the whole gene. So basically look at it as a library. Do you want to go to, back in the days we used to go to a library and scan every single book and then find our own book. Nowadays you just type whatever you're looking for and the system will find it for you, right? Or you basically Google one information and instead of searching the whole internet, you just Google something and then you find your query result. So basically this was the idea. Can we actually use CRISPR in some sort of a sensing platform where we can look for our target gene? So this is what we did. We combined biosensors with, we combined the transistor with this CRISPR molecule. So the way it worked I moved a lot of my slides last minute, but the way it works now, we have a transistor that tells us things that are happening at its surface, right? We put this enzyme on the surface of this transistor, and every time this enzyme searches and finds the target, it just brings the DNA down. And that DNA, when it comes down, it changes our transistor's property. And so we use it for our DNA detection. So what we did with this, without amplification, because our transistors were very sensitive, without amplification, we were able to detect a few mutations in human genome. So we looked for a mutation in people who have Duchenne muscular dystrophy. So this is a disorder that basically the muscle gets weak and the dystrophin protein is not functional. So then the patients, they actually start, like the muscle strength, they lose their muscle strength, and they usually it happens in boys, and then they die usually in their 20s. So what we did, we basically just took the sample from this patient and looked for two specific mutations that was very common in this patient. And we programmed the CRISPR to actually just go detect those two regions. So then we just turned on our transistor and we put the DNA on the transistor, and the CRISPR started searching for its target. And then once it found the mutation, the target, it just basically binds to it, and then you can see the result. So again, the vision was, can we actually use this searching capability of this very, very powerful technology for something that is beyond gene editing? That's what most of the people who are using CRISPR are focusing on. So I think we have
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location.
I'm just gonna go to this one where it talks about the system that we developed. It shows you that
this small chip has the power to detect genetic mutations that cause disease and can help doctors provide better treatments, all within minutes.
Let's say you have a DNA sample from a patient and you want to make sure that that gene that you're interested in is a healthy gene. You introduce that to our device, Crispr Chip, and then Crispr Chip will start looking for the DNA.
Crispr Chip is a handheld device that combines thousands of CRISPR molecules with a graphene transistor. Researchers designed CRISPR to scan through DNA and find specific genes or mutations, while the transistor is super sensitive to electrically charged materials, like our DNA.
You introduce the DNA from the patient on top of the graphene surface in Crispr Chip. The CRISPR scans these DNAs. If CRISPR doesn't find its target gene, it won't bind to it, and the DNA is released. But if it scans and it finds its target, then it will bind to it. And this binding will create an additional charge on top of the graphene surface, and this can be sensed with Crispr Chip. This shows you the response that we get from Crispr Chip. Here, you're looking at when we actually introduce our DNA sample and remove it from the sensor. This endpoint, if it doesn't match this initial point, that means that our Crispr Chip was able to detect gene that we programmed it to detect. This is which sample? This is a DMD sample. So far we have tested the technology for muscular dystrophy. We are currently testing it for sickle cell disease, which is much more difficult to detect. And we are hoping to increase the sensitivity of our system to detect infectious disease as well. Our system is a few years away from being product for diagnostics, but in its current format, it can be used for many other applications. For example, it can help scientists and researchers design better CRISPR complexes when they're trying to use these complexes for gene editing.
Clinical trials are using CRISPR to edit and hopefully cure genetic disease. But researchers must first test whether the CRISPR molecule they design will find and edit the mutated gene and not a healthy gene nearby. With Crispr Chip, they can test this faster than ever before. In the future,
Okay. It's 4:15, so I think I have to wrap up. But so one of the main important things that we have to consider is that we go next. Okay, one of the first things that we have to consider that we're passing around the chip that we actually made the CRISPR chip possible. So when I started this project, I was using these chips. I was making like wires, and it was a messy platform. But then through collaboration with our industrial partner, which is sitting there, the person who actually invented that and created that commercial chip is sitting right there. He was supposed to tell me the story, but we're short in time. And he was the first person to actually take graphene and make a commercial chip out of it from his basement to now to a company that it's big, and we're hoping to become a part of it. So now in collaboration with them, we're trying to commercialize this not only for diagnostic application, but just to help people who are developing therapeutics, gene editing application from this CRISPR technology, to make sure that the gene, that the complex that they're designing, it's targeting the right site, it's not cutting different region, it's the most efficient one for that specific patient. So we're really hoping that we can advance the technology in that aspect. So again, my vision is Googling your genome. We are Control F right now, but then we'll become a Google hopefully in the next 20 years or while I'm alive, hopefully. Thank you so much. Any questions?